Hi, this is Alex from devstories.eu. AlphaFold, Protein Structure Prediction, Episode 1 of 21. For 50 years, scientists could read the raw text of our DNA, but they could not predict what that text would actually build. They had the source code of life, but no idea how it compiled into the physical machines running our cells. This grand challenge is known as the protein folding problem. Proteins are the molecular machines that keep you alive. They handle almost everything happening inside your body, from carrying oxygen in your blood to fighting off infections. Every protein begins its life as a one-dimensional string. This string is built from a vocabulary of 20 different chemical building blocks called amino acids. Think of the amino acid sequence as the raw source code of biology. It is a linear string of characters read sequentially. But a straight line of amino acids cannot do any actual work. Just as a plain text file needs to be compiled into an executable binary to run, the one-dimensional amino acid sequence must fold into a highly specific three-dimensional shape. In the biological world, shape dictates function completely. The physical contours of a protein determine what it can interact with. A protein folded into a pocket shape might catch and break down a specific sugar molecule. A protein folded into a rigid tube might act as a structural support for a cell. If the folding process goes wrong, the biological program crashes. In humans, misfolded proteins are the root cause of many severe diseases. The sequence of amino acids contains all the instructions needed to form this exact three-dimensional structure. The different amino acids have different chemical properties. Some carry positive or negative charges and act like magnets. Some repel water and try to hide in the center of the structure, while others are attracted to water and pushed to the outside. These competing physical forces cause the string to tangle, twist, and snap into a single, stable configuration. Here is the key insight. The math behind this folding process is staggering. A typical protein chain is made of hundreds of amino acids. The number of possible ways a chain that long could bend is around 10 to the power of 300. A scientist named Cyrus Leventhal pointed out that if a protein tried every possible shape sequentially to find the right one, the process would take longer than the age of the universe. Yet, inside your cells, a new protein string snaps into its correct shape in a few milliseconds. The protein folding problem is the attempt to bridge this gap. It is the challenge of taking a one-dimensional amino acid sequence as the only input and computationally predicting its final three-dimensional structure. Historically, scientists had to rely on slow, physical laboratory techniques to map these structures. Methods like X-ray crystallography involved freezing proteins into crystals and firing beams at them to measure the angles of the scattered light. Finding the structure of a single protein could take years of painstaking trial and error. Because genetic sequencing technology outpaced physical mapping, the scientific community accumulated hundreds of millions of known one-dimensional sequences, but mapped the 3D structures for only a tiny fraction of them. We had endless lines of source code, but no decompiler to show us the executing logic. Solving the protein folding problem computationally gives us the exact mechanical blueprints of biology, turning drug discovery from a slow process of laboratory guessing into precise, targeted engineering. If you enjoy the podcast and want to support the show, you can search for DevStoriesEU on Patreon. That is all for this one. Thanks for listening and keep building.